Hello friends, today we are going to study about composition, structure and function of carbohydrates part 9. In this part, we'll study about glycoproteins. In the last video, we studied about proteoglycans. Now we will study about glycoproteins. What is the difference between proteoglycan and glycoprotein? Both are the combination of protein and carbohydrate. That is proteoglycan and glycoprotein. Both are the combination of protein and carbohydrate. But in case of proteoglycan, the combination is of protein with glycosaminoglycans. Whereas in glycoprotein, the combination is with protein and oligosaccharides. Some glycoproteins have a single oligosaccharide chain, but mostly they consist of more than one oligosaccharide chain. So the difference between glycoprotein and proteoglycans, apart from the carbohydrate type, another difference is the carbohydrates are smaller and structurally diverse in glycoprotein compared to that with proteoglycans. Now the carbohydrate which is attached with the protein in case of glycoproteins, that carbohydrate is either O-linked or N-linked. What is O-linked? In case of O-linked, the anomeric carbon of the carbohydrate is attached with serine or threonine. If you see the structure here, this is a carbohydrate, this is a protein. So the anomeric carbon, the OH attached with the anomeric carbon is linked with the serine residue. So this is O-linked because it is linked via oxygen O. Another is N-linked. What is N-linked? In case of N-linked, if you see the structure here, this is a carbohydrate. This is the asparagine residue. Now how it is linked? The N, that is the amide N of the asparagine is linked with carbohydrate. And what this bond is? N-linked glycosidic bond. Okay, so carbohydrate is attached with protein, either O-linked or N-linked. In case of O-linked, it is serine or threonine to which the carbohydrate is attached. If it is N-linked, it is asparagine to which the carbohydrate is attached. These glycoproteins are found on the external surface of the plasma membrane. So the well-characterized glycoprotein is a glycophorin A. What is a glycophorin A? It is a glycoprotein which is found in the erythrocyte membrane. The carbohydrate constitute the 60% of glycophorin by mass. Total 16 oligosaccharide chains are attached with the protein, among which 15 oligosaccharide chains are O-linked and one is N-linked. Apart from glycophorin A, the other examples of glycoproteins are antibodies, follicle stimulating hormone, luteinizing hormone, thyroid stimulating hormone, lactalbumin, ribonuclease. These are all examples of glycoproteins. Mostly the proteins which are secreted in the blood, they are mostly of glycoprotein nature. The oligosaccharide which is attached with the protein. Now the same protein, if it is found in two different types of tissues, the pattern of glycosylation will be different. Suppose there are only one protein, protein 1. This protein is found in some A tissue and the same protein is found in some B tissue. In both the cases, the glycosylation pattern, the carbohydrate which is attached to this, that will be different. The glycosylation pattern will be different. So hence, the glycosylation pattern or the oligosaccharides which are attached with the protein can act as a marker. They can act as a tissue-specific marker because they are different in different tissues. The same protein found in different tissues will have different glycosylation pattern or different oligosaccharides attached with it. Therefore, the oligosaccharide chain represent a tissue-specific marker. Now, what are the function of these oligosaccharides which are attached with the protein? So, they are soluble, they can form bond with water. Therefore, because of their polarity, they can alter the solubility of the protein to which it is attached. Next is protein folding. The oligosaccharides are attached with protein in endoplasmic reticulum and Golgi complex. This attachment of oligosaccharide with protein determine the tertiary structure of a protein. So it helps in the protein folding. Also, the carbohydrate containing many negative charge, what will happen? If many negative charges are present, they will repel each other. Because of this repulsion, the protein will gain a structure. What kind of structure it will gain? Extended rod-like structure. So, the carbohydrates or the oligosaccharides attached with the protein can determine the structure or the folding of the protein. Next is protection from proteolytic enzyme. Because the carbohydrates are bulky and they have a negative charge. Therefore, it can protect the protein from many proteolytic enzymes. These are few functions of the oligosaccharides which are present with protein. We'll study some other functions in the next part of the carbohydrate video. Next, we come to glycolipids and lipopolysaccharide. These are what? The carbohydrate attached with lipid molecule. So here, carbohydrates are covalently attached to lipid molecule. Here also, the oligosaccharides are attached to the outer surface of the membrane. One of the best example of glycolipid is ganglioside. These are membrane lipids of eukaryotic cell. It contains complex oligosaccharide which contains sialic acid and other monosaccharide residue. These are mostly found on the neural cells and also they are found in other cell types. So ganglioside is one of the example of glycolipids found in eukaryotic cell. Lipopolysaccharides are found in bacteria. So the gram-negative bacteria such as E. coli and Salmonella typhimurium, they contain lipopolysaccharides on their outer membrane. Now, these lipopolysaccharides act as a prime target for the antibodies. Whenever these gram-negative bacteria attack us, our immune response will produce antibodies to protect ourselves. And those antibodies are targeted for the lipopolysaccharide. Hence, the lipopolysaccharide can use for serotyping, for the identification of these two particular microorganisms. The lipopolysaccharide, which is attached with typhimurium, it contains 6 fatty acid which is bound to 2 glucosamine residue. And to 1 glucosamine residue, complex oligosaccharides are attached. The lipopolysaccharides of some bacteria are toxic to humans, because of which when these bacterial infection occur to us, we experience toxic shock syndrome. Okay, so these toxic shock syndrome results when our gram-negative bacteria cause infection to our body. So now one question from my end for you all. You have to tell me the difference between glycolipid and lipopolysaccharides. So what is the difference between glycolipid and lipopolysaccharide? Both are the combination of carbohydrate with lipid. So you have to tell me the difference between both. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.